Good morning, students. Today we'll have our ninth session in microbiology, and the title of this session is Bacterial Identification and Classification. This will be the last session in bacteriology. Then we will be moving to virology and mycology. But in this session, we will be defining and explaining bacterial identification, how to be identified, and classification. And we already explained the triangle of the taxonomy that includes identification, classification, and today we are going to concentrate on identification and classification. Bacterial identification, this is the methods used to identify the genera and species of isolated bacteria. The source of the bacteria could be a clinical sample, for example, blood, urine, or fecal sample, or the source of the sample of the bacteria could be from the environment, soil, water, air. So there are different samples, but at the end, you will be isolating the bacteria in a culture media. So you want to identify this bacteria using different techniques used in the microbiology lab. So you need to identify to know the genera of this bacteria and the species of this bacteria. So there are different and several methods used for bacterial identification. The first method is cultural methods for bacterial identification. So in this method, as we explained before, culture, that means already the bacteria is growing in a solid medium. So when we say a culture, that means we are speaking about colonies of a bacteria. And we already defined a colony. A colony is a gross, or that is the gross of a number of bacterial cells in the form of a single unit that is started from a single bacterial cell. So a colony defines similar bacterial cells from the same genera and from the same species, growing from the same single cell of a bacterial cell in a culture medium. So one of the identification methods is identifying the characteristics of a colony. So from this, we need to look onto the culture media, which is usually a agar, solid medium, on the petri dish, to look into the shape of the colonies. Is it circular? Is it regular, irregular, or is it rhizoid? So there are different shapes of bacterial colony. We are going to have a separate microbiology lab about the characteristics of colonies. The other type, the other way or characteristic we need to look onto the cell, the colony, bacterial colony, is the size of the colony. Is it small? Is it medium? Or is it large? And then we have to note if it is pigmented, if it has a color. Is it gray? Is it pink? Is it green? So does it have a pigment or is it just white? The elevation of the colony, you need to note the elevation of the colony to look into the side of the petri dish and note the elevation of the colony. Is it flat? Is it elevated? Is it convex? Is it concave? So you need to know the elevation of the colony. The fifth characteristic of a colony is the margins of the colonies. Are they smooth, rough, or wavy? So there are different types of margins for the colonies, and this is a characteristic of the growth of bacterial species. The second way or method of identifying bacteria is the staining and microscopy for bacterial identification. And in this method, we already defined before the gram stain. So the gram stain is one of the most important step to identify a bacterial cell. So you prepare the slide from as you already trained on that in the previous microbiology lab, how to stain a bacterial cells on the slide. So you stain the bacterial cell using the steps of gram stain, and then you observe it under the microscope, light microscope, and you define the result. There are two results: either gram positive cells, bacterial cells, or gram negative cells. So this is the basic step in identifying a bacteria, because using gram stain you will be able to identify what type of cell wall does these cells have. So but gram stain is a very step in identifying bacterial cells. There are other types of stains, which are simple stain. If you want just to define the shape of the cell, is it cocci, or is it spiral, or is it um, 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 bacillus, regardless of what type of cell wall, then you can just stain. A simple stain is just staining a bacterial cell with one stain, not a complex stain um, like gram stain. So if you just stain it with crystal violet, this is what we call a simple stain. So you can use simple stain. It depends on what you want to study on the bacterial to identify the bacteria. So there are different type of other specific stains like flagellary stain, endospore stain, and these are a range of staining techniques used in different microbiology laboratories for different purposes. But the most important here we need to concentrate on gram stain. So you can use different, since you have different staining techniques, definitely you will be using different microscopies like light microscopy, fluorescent microscopy. At this level you don't need to know more about this microscopy, because they are used for different purposes. But the basic microscopy we use in the microbiology is the light microscopy, as we explained in the first microbiology lab. So these are the different microscopes. There is the electron microscopy. Of course, this is used more if you want to study more in depth, or higher magnification, very higher magnification of certain parts of a bacterial cell, like a flagella or pili, or organelles, or I mean inclusion granules inside the cell, and so on. So this is a very high magnification used for other purposes of studies. So usually in the bacteria identification using staining and microscopy techniques, um, usually in the lab they use what we call a dichotomous key for identifying or classifying the bacteria. This is a very simple tool that uses two characteristics of a bacteria. One characteristic is the staining result, and the other characteristic is the shape of the bacterial cell, if it is cocci, if it is bacilli, or spiral, or maybe not an irregular shape. So you want, these are the two characteristics: the shape and the gram stain result. So this dichotomous key, it's a tool. As I mentioned, it is a tool, can be defined by based on these two characteristics, and used in the lab just to to further move on with identification. As you move on with identification, then you will be able to narrow down the classification or identification of the bacterial genera or species. For example, if we take the first one, if it is gram positive bacteria, then you go to two. Then you go to two and see if it is is it a cocci. Then you call this bacteria it is gram positive cocci. If it is not a cocci, then you go to four. You go to four and identify the shape. So if it is a bacilli, then this is gram negative bacilli. If it is not bacilli, then you go to six. And then if it is club shaped, then it is identified as corynebacteria. If it is not, then if it is irregular, then it is propionobacteria, and so on. This is how we use this dichotomous key. Dichotomous, that means two characteristics. Di, that means two, and when you say cotomous, that means you are using two characteristics of an organism or bacteria cell to define or identify the type of bacteria. From this, then you can move on to the third level of identification, that is used by chemical tests for bacterial identification. In the biochemical test, this is a method based on the um, identifying the bacteria based on the types of enzymes in the bacterial cells. And this type of enzymes definitely define the physiology and metabolic activities that is taken or undertaken by the different types of bacteria. And the physiology and the metabolic characteristics, as we explained before, the nutrition, the oxygen requirements, the fermentation of sugars, these are all physiological and metabolic activities. So they differ from one genera to the other and from one species to the other of the bacterial cells. So we will define different types of biochemical tests and see how this will identify the bacteria based on their physiological and metabolic activities. In the biochemical test used for bacterial identification, we are going to concentrate on the common biochemical tests used in the lab. Of course, there are very large number of biochemical tests. Some of them are very specific to certain type of bacteria, and but usually in the lab they start, especially if you don't know any background about the bacteria, they start with the common biochemical test. So in this session we are just going to define the common biochemical test, just to get oriented about how this test define or give results to identify the bacteria. These tests are, we call them qualitative tests because they just give reactions. They don't give quantity. They don't depend on the quantity or the number of bacterial cells in a medium. No, it defines the physiology and the metabolic process of a bacteria. What type of enzymes this bacteria does it have, regardless of the number of bacteria in the that's growing in the medium. So these are qualitative. We call them qualitative tests. It's either positive result or a negative result. The first test we are going to explain is the indole test, and the indole test is actually testing the ability of the bacteria to utilize the amino acid tryptophan. And tryptophan is one of the amino acids that is essential for human beings. So this test is usually used to identify the members or differentiate the members of the family Enterobacteriaceae. Enterobacteriaceae is a family of bacteria that that have a number of genera and number of species, different types of species, but they are all um, their main habitat is the gut or the intestine of the human beings and animals. So these bacteria, they use the if they have a certain type of enzyme in their cells, which is tryptophanase, they have the ability to hydrolyze and metabolize the amino acid tryptophan. The metabolic product of this hydrolysis is a chemical known as indole. So if you grow this bacteria in a medium that contains this essential amino acid tryptophan, if the bacteria has this enzyme tryptophanase, then it will hydrolyze the tryptophan and producing the chemical indole as a byproduct of this reaction. So if you want to detect this indole, then there is a specific indicator or reagent. We add it to this medium, and this reagent is actually testing the change in the pH of the medium. So if indole is produced, once you add this reagent, which is a type of indicator, pH indicator, it it turns to give a ring because the indole in the broth medium, where there is a tryptophan and the bacteria has tryptophanase, if if it utilizes or hydrolyzes the tryptophan, it produces the indole. Once you add this reagent to the tube, it gives a ring on the top of the tube, on the top of the medium, and this shows that the bacteria utilize the tryptophan and produce the indole. If it doesn't have this tryptophanase, the enzyme, then there will be no production of indole. The bacteria will just grow without hydrolyzing the tryptophan. So this is a qualitative test identifying between the different types of Enterobacteriaceae family, between indole positive and indole negative. So that's why we call it indole test. The other type of test is the methyl red Vox Proscure test. So the MRVP test is used um, is used to test the to differentiate between the types of bacteria that utilizes or hydrolyzes glucose either aerobically or anaerobically. So, and this one and this and the fermentation of this glucose, we already defined it before. There are different pathways, either aerobic, and this we call it a pathway which is a mixed acid fermentation pathway. So there is a production of different types of acid from the hydrolysis or the fermentation of glucose. The other pathway is the what we call the the butanediol fermentation pathway, and this is producing acetone byproducts as acetone or 2,3-butanediol. So this is our different pathway. The first pathway, because producing acid, this we call it an aerobic pathway, and the other one is non-aerobic pathway, and we call it butanediol fermentation pathway. So we'll be differentiating between how the bacteria ferment the glucose, either aerobically or anaerobically. Aerobically, it produces acids, and anaerobically, it produces acetone and other organic compounds. So if the bacteria, we usually grow the bacteria in the medium, and there are, for each test there is a specific, of course, medium. So we grow this bacteria into the medium, and the medium is called MRVP, or methyl red box proscure broth, and then incubated overnight, which is more than 24 hours or 24 hours. So following incubation, then we add the pH indicator methyl red, and if the medium, the broth change, as you see here, there is a ring, the red ring, then this is, we call it methyl red positive. Methyl red positive because it ferments glucose aerobically, and that's why it's producing acid. And this, the production of the acid, once react with the pH indicator methyl red, it turns into red color in the medium at the ring, as you can see here. So this one, we call it methyl red positive, that ferments glucose aerobically, producing acids. And this is methyl red negative, that means it doesn't hydrolyze or ferment glucose aerobically. Since it doesn't ferment aerobically, then we have to detect for Vox proscare. So we detect for Vox proscare by adding a specific reagents. We call it VP1 and VP2, and this is added to the median, and if it gives like a ring on the broth, and this is a red pink to cherry colored color, and this indicates a positive box proscare test. That means the bacteria ferment glucose anaerobically. So MRVP test differentiate between the bacteria that ferment glucose aerobically and anaerobically. The third type of test is the citrate test, and this is used to detect or to differentiate between the bacteria that uses citrate as a carbon source. So we grow the bacteria in a medium that contains citrate, and this medium is called Simon citrate agar. And if it uses the carbon, then there will be a change in the color, as you can see here. Citrate test. There is a change. This is a slant in the tube. As you can see here, there is a change in the color. Once it grows, the color of the medium is green. If it utilizes the carbon, then the color of the medium change into blue. So this is what we call citrate test. Testing the bacteria if it utilizes the citrate. Citrate, as you know, it is a type of um, a chemical that contains carbon source. So it utilizes this citrate as a source of carbon, and the medium we grow the bacteria on, we call it Simon citrate agar. So if it change into blue, then this means this bacteria utilizes citrate for as a carbon source, and that's why the medium changes its pH, and the indicator in the medium changes to blue because it becomes more alkaline. The other test is the urease test, and this is used to detect if the bacteria has the enzyme urease, and this is usually the urease test is by growing the bacteria in a urea agar, and this is a type of medium that contains agar and all the other nutrients, and the urea as an organic component is added to the media. We call this medium as Christine's urea agar, and usually in the form of a slant in a tube. And then we grow the bacteria, we inoculate the medium and incubate it overnight. And we read the result based on the change in the color of the medium. Usually the medium color is yellow, okay, and it has because the medium itself it has a pH indicator. So if the urease enzyme is found in the bacteria, it hydrolyzes the urea in the medium and change the urea into ammonia and carbon dioxide. So once it is changed into ammonia, then the pH of the medium changes to be alkaline, and the medium changes from yellow color into red. There are different levels of the change of the medium. If we change the whole slant or the whole tube, as you can see here, the whole tube turned into pink or red color, then this is shows that the bacteria is positive. That means it contains an enzyme urease that hydrolyzes the urea in the medium. If it is only partially changing the medium change into partially red, for example, in the only the slant part, then this we call it delayed positive bacteria. That means it has the enzyme, but it takes time to hydrolyze the. It's not very active as the pure positive bacteria. So this is we call it. It has the enzyme, but we call it delayed positive result. If it doesn't have a urease enzyme, then the medium remains as it is in the color. Its color remains as it is, so it remains as yellow or as the medium original color. So this one we call it negative urease test. That means it doesn't have the enzyme urease in the bacterial cell. The fifth type of biochemical test used in the lab is what we call the triple sugar ion test. From its name, triple sugar, that means this medium contains three types of sugars, which is glucose, lactose, and sucrose. Beside, this medium also contains ferrous ammonium sulfate, and this medium is used to test the bacteria if it has ability to hydrolyze or ferment glucose, lactose, and sucrose, beside producing hydrogen sulfide from ferrous ammonium sulfate. And if you remember when we spoke about the ability of the bacteria to utilize ammonia and ferrous salts for as electron donors, or as source of hydrogen, or source of carbon dioxide, hydrogen sulfide from utilizing ferrous as a metal in the metabolic process of the bacterial cell. And usually the byproduct is hydrogen sulfide, is a gas hydrogen sulfide. So this medium triple sugar ion agar is testing two things: testing the hydrolysis or the fermentation of the three sugars, if it has the ability to ferment the three sugars or one of them, or maybe two of them, and then we'll see how the results comes out. And the other thing is the production of hydrogen sulfide from ferrous ammonium sulfate. That means it is using this salt as a source of sulfur and as a source of electron donor and as a source of ferrous, or metal, the metal ferric. Um, so in the medium is usually is in the form of a slant. That means in a test tube, it has it has it has two parts in the test tube. What we call the butt. The butt is the lower part of the medium, which is filling the tube, and the slant is the curved or the slopy part of the medium. So this is we call it a butt, and this is we call it a slant. So you inoculate the medium with the bacteria you want to identify, and then you incubate it overnight. And so this medium is called triple sugar iron agar. So and then you incubate it overnight. Next day, you after the incubation, that means you incubate it in the right temperature. When we say incubation, that means we incubate the test tubes in the optimum temperature that is required by the bacteria. All or 90%, more than 90% of the pathogenic bacteria that cause disease, they their optimum temperature, incubation temperature, is 37 degrees centigrade. So you incubate the bacteria in the incubator, and then you read the result the next day. So the result is either one of these results. Either there is no change in the medium, there is growth, but there is no change in the color or the texture of the medium, and this is, we call it negative, or sometimes we put a control, of course. That means the bacteria does not hydrolyze, does not utilize the sugars in the medium, nor the ferrous ammonium sulfate salt. If the medium produces a red color, a reddish color or pinkish color, change from yellow, from red to yellow. That means from red, usually the medium is reddish in color. If it change from red to yellow, that means in the slant only. That means the bacteria has the ability to ferment or utilize glucose. If the medium change in the slant and the bud, both of them, then that means the bacteria has the ability to ferment the three sugars: glucose, lactose, and sucrose. If there is production of sometimes there is, of course, it depends on the type of fermentation. There is a crack in the medium. That means there is a production of carbon dioxide, and this is an indication there is fermentation, which is aerobic fermentation. If there is a change in the color to black, if there is a black color, that means the bacteria can utilize ferrous ammonium sulfate as a source of sulfur, as a source of electrons. And so this is oxidation reduction reaction, and there is a production of hydrogen sulfide, and this changes the color into black, and there usually be crack in the medium, the agar medium, um, it, like cracks in the medium, because there is production of hydrogen sulfides that crack the the medium within the test tube. But usually the most positive result for production of or reduction of ferrous into ferric is the dark black color, which is coincides or reflect the utilization of the salt ferrous ammonium sulfate and production of hydrogen sulfide. So this is how you read the result, and this is one of the most useful biochemical tests, because you will be able to test the ability of the bacteria against three types of utilization of three types of sugars and a salt, which is ferrous ammonium sulfate. And this is widely used in the microbiology labs. The sixth test is the slide catalase test, and in this test, we are testing if the bacteria has the enzyme catalase. The enzyme catalase, it is usually used by the aerobic bacteria that utilizes oxygen as a source of um, of respiration, and and utilize it in other types of metabolic activities within cells. We call it a slide test because we take a slide, a glass slide, and put a loop or part of the bacterial cells in the slide, and then we add the chemical hydrogen peroxide. As we know, hydrogen peroxide, it contains oxygen and hydrogen. So if the bacteria has the catalase enzyme, it hydrolyzes the hydrogen peroxide. Hydrogen peroxide, as you know, it's a liquid form, and we add a drop to the bacterial cells on the slide. And if the bacteria has the catalase enzyme, then it hydrolyzes the hydrogen peroxide into oxygen and water. So you will see the bubbles of oxygen rising from the bacterial cells on the slide. That is why we call this is a positive catalase. That means it has the catalase enzyme. That means it is an aerobic bacteria. And the other one, it doesn't show any bubbles after adding the hydrogen peroxide. That means it is negative. It doesn't have the catalase enzyme, and this is indicates that the bacteria either anaerobic or microphilic that requires carbon dioxide. So this one of the tests used to detect to identify the bacteria by the position or the if the bacteria possesses or has the catalase enzyme. The other type, number seven, of the of the biochemical test is the oxidase test. An oxidase test is, as its name indicates, is testing the bacteria that possess or have the enzyme oxidase. The enzyme oxidase is one of the enzymes that the bacteria produces to be able to utilize the oxygen that is produced from the different biochemical reactions within the cell or metabolic processes within the cells for as the result of oxidation reduction reactions. So the oxidase helps the bacteria to utilize the oxygen for other as an actor acceptor in aerobic respiration. So usually we do this test in the lab using a filter paper, and then we add a small part or inoculum, small inoculum of the bacterial culture onto the paper, as you can see here. And then we add to this bacterial culture one drop of the oxidase reagent. This is a specific reagent that detects if the bacteria has the oxidase enzyme or not. So if the color of the culture or the inoculum on the filter paper change into a purple color within 20 seconds, very fast, that means the oxidase, the bacteria is oxidase positive. If it didn't change, that means it is negative. The reaction should be very fast within 20 seconds, because otherwise if it change after more than 20 seconds, then this is a false positive result, because the reagent itself, it is oxidized by the oxygen in the air. That is why the reaction should be very fast once you add the drop. If it changes within 20 seconds, that means the bacteria has the oxidase enzyme. So this is how you detect for the oxidase enzyme in the bacterial cells, which help in the aerobic respiration of the bacteria, utilizing the oxygen that is produced from the different metabolic activities or processes within the cell from the reduction and oxidation reactions within the cell. The eighth type of biochemical test, what we call the sugar fermentation test. And this is actually used if you want to detect the bacteria has the ability to ferment sugars in general, regardless if it is glucose, sucrose, or cellulose, complex sugars, and so on. So this bacteria, so this test we use a broth called carbohydrate broth. We call it carbohydrate broth because it contains a very large number of sugars, different types of sugars, monosaccharides, disaccharides, and cellulose, um, different types, mannose, and so on, xylose. And so these are a combination of large number of carbohydrates in a single medium. So if you want to detect if this bacteria has ability to ferment or utilize carbohydrates, and you can even differentiate between aerobic and anaerobic fermentation. So in this test, we prepare the broth, the carbohydrate broth. That means it is a liquid medium, and we put it in tubes. And usually the color of the medium is red. This is a normal color of the medium. It has an indicator, pH indicator. Of course, if it change, if it change the if there is a production of different byproducts from the fermentation, then the color of the medium changes into yellow because there is a pH indicator within the medium. So we incubate after we inoculate the bacteria the broth with the bacteria. We incubate it for at 35 at 37, usually 35 to 37 degrees centigrade, for 18 to 24 hours in the incubator. And then we observe the color of the broth. And inside this broth, we usually put a small tube. This small tube is inverted within the large tube within the broth. We use this tube and we call it Durham's tube. Durham's tube is smaller test tubes inserted upside down in the test tube. And this just to detect if there is a production of gas from the fermentation process. Because with the production of gas, you will be able to determine if this bacteria ferments the sugars within the carbohydrate broth aerobically or anaerobically. And usually the gas bubbles appear within this tube. So for reading the result, if the medium color change into yellow without production of gas, this also we call it positive fermentation of carbohydrates. And then if it change into yellow, but there is the gas bubbles within the Durham's tube, the small test tubes inside the medium, then this is positive fermentation again with the production of gas. And usually in this case, it is carbon dioxide production. So we can differentiate between aerobic and anaerobic fermentation of sugars. So this is the one of the most highly used tests within the microbiology lab. There are other types of tests, as I mentioned before. They are very specific to certain types of bacteria. If you manage to identify with the common test the characteristics of this bacteria, the enzymes that's contained within this bacteria, you can further move on with the specific test like DNA test, nitrate, if they have the ability to utilize nitrogen from nitrate, like the coagulase test, if you want to detect if this bacteria, like Staphylococcus, they use the coagulase test to detect if it has ability has the enzyme coagulase to clot certain types of proteins, like the test that we already explained before, hemolysis test in the blood agar. And we already defined before in the previous session that if you grow the bacteria in a blood agar, for example, it can depends on the metabolic process within the bacteria and the type of enzymes, if it either produce a beta hemolysis, alpha hemolysis, or gamma hemolysis. So these different tests, I'm just listing here some of them to show that there are different other types of tests, but they are more specific for further identification of the bacteria after performing the common tests we already explained. So the microbiology identification of bacteria, we already defined the 8 common tests, and the ninth is a collection of tests specific for certain types of bacteria. Different labs use different flow charts. Flow charts are type of charts or processes that follow certain pattern to identify bacteria. So it depends on the lab how they follow, and their protocol defines their protocol of work within the microbiology lab. Usually, these flowcharts are found in research labs or in the medical diagnosis labs for identifying the bacteria. Uh, these labs, they put like flowcharts, as you can see here, an example of a flowchart from one of the labs. If they have an unknown bacteria, they first stain the bacteria and define if it is gram positive or gram positive or gram negative, and then define if it is cocci or bacilli, and then they perform catalase, and then if it's catalase negative, they move on to a specific biochemical test, a clean test, to identify if it is positive or negative, and then from this they can determine if it is the genera of the bacteria, and if it is catalase positive, they move on to define the growth of the bacteria, to further identify the genera. And so this is a type of flow chart. As you can see, there are different tests, biochemical tests. They follow this biochemical test depending on what is coming out of the result of the gram stain and the shape, the dichotomous key we already explained before, if it is gram negative or if it's a cocci or if it is a bacilli, and so on, according to the stain result and the shape of the bacteria. Then this determines further biochemical tests to be done to identify the genera of the bacteria. These protocols or flowcharts are usually an extract or determined by using a well-known manual that is already developed and published. We call it Berg's Manual of Systematic Bacteriology, and this manual, it is found in all bacteriology labs for identifying the prokaryotic organisms, archaea, and bacteria, emphasizing to the level of bacterial identification to the level of species, and using different characteristic attributes, the gram stain, the biochemical test, the microscopy results, um, gross on the culture, the culture characteristics on the medium, and so on. So it usually, the labs, they develop these flow charts from this manual just to help them to process the samples in a faster rate, and it is very guiding process because the manual, it has almost 5 volumes, very big volumes, and by experiencing the process of identifying bacteria, you will be able to define a flow chart to follow in the lab so that you can identify the bacteria, especially the pathogenic bacteria that is causing disease. And usually these flow charts are followed, as I mentioned before, in most medical labs for diagnosis of diseases, and in research labs for conducting further studies in bacteriology. We move on to bacteria classification, as we mentioned in the identification section, that identifying bacterial species depends on Berg's manual. And actually, Berg's manual was first published in 1984, the first edition, and it's called Berg's Manual of Determinative Bacteriology. And in this edition, the and actually this manual, it is of course developed by eminent and expert scientists in bacteriology, and they classified the kingdom Prokaryota at that time into four divisions: Gracilicutes, and this is they define these divisions based on this gram staining, presence of endospores, general shape, motility, morphology, and mode of energy production. And as you can see, these divisions are based on phenotypic characteristics. That means the general shape of the bacteria rather than by chemical or enzymes or metabolic activities. So they classify the kingdom prokaryote into four divisions. Grassicutes, they have gram negative cell wall, and firmicutes, they have gram positive cell wall. Tourniquets, they do not have a cell wall, and this is like mycoplasma. And we mentioned before that there are certain type of bacteria which have irregular shape because they don't have a cell wall. And mendoscutes, they lack peptidoglycan in their cell wall, and they are similar to archaea. So these four divisions were in the first edition of the manual. However, in the second edition, of course, the science of bacteriology advanced, and the studies of molecular biology, studying the DNA and RNA and the protein content of the cell, became the tools and the techniques became more advanced. That's why they include it in the second edition, and they call it Berg's Manual of Systematic Bacteriology. Because they moved on into the genetic part of the bacteria, and they use what is called phylogenetic characteristics such as DNA, RNA, and proteins. And when we say phylogenetic, that means we are comparing the similarities and differences among or across the bacterial species based on characteristics of species related to DNA, RNA, and they interpret their evolution relationship and origins. They go back to their origins based on the DNA, the source of the DNA, the structure of the DNA and the RNA, and of course, if you study the DNA, you will be able to define the protein content of the bacterial cell. So there are two at that time. There are two editions. The first edition is the Bergsman Determinative Bacteriology, and the second edition is changed because they moved into more depths into the metabolic activities and the genetic content of the bacteria. They call it systematic bacteriology. The last edition is the ninth edition, and this is published in 1994, and this consists of 5 volumes. And as I mentioned, this manual helps in the identification of the bacteria based on different uh, laboratory techniques, including biochemical, morphological, genetic content, serological content, that means antigen antibody reactions, and so on. So, in this latest edition, which is the ninth edition, it's divided into two domains, archaea and bacteria. The domain archaea contains 2 phyla, and the domain bacteria contains 23 phyla. When you say phyla, that means below the phyla there is genera, there is species, and so the manual helps you to identify the phyla first, and then the domain. According to the domain, you can identify the phylum, and then from the phylum you can identify the different downward classification of the bacteria up to the species level. I'm just putting here um, like shots or copies of the manual just to show the phyla. But of course, these are the different volumes, very large volumes, and usually found in most of the in all of the bacteriology labs to help in the identification of the bacteria. This is just to know the different phyla, and just not for you to memorize this. It's just to know that these volumes have different this manual have different volumes, and in each volume they're addressing different types of bacteria, and the different phylum, and under the phylum there are different identification processes leading to the species identification of the unknown bacteria. Out of the bacteriology we all studied, I'm just going to concentrate on the bacteria species of medical importance, because you are health science students. You need to know what type of bacteria that causes what type of disease, and what is the reservoir of this bacteria, where are they found, the morphology of this bacteria. And this is very important. You need to note, and I put here some tables, three tables, very important. You need to know, and know by heart. And this is the species that you need to know exactly what are they, and how and what type of disease they cause. So bacterial species of medical importance, usually there are two types of bacteria, either commensals, and this is the type of bacteria we call them microbiota, or in general the science that deal with these commensals that are found. This is a bacteria that found normally in the human body, either in the skin, on the skin, or in the mouth, or in the gut. These are bacteria that live symbiotically. That means it benefits the human body or the animal body, and the same time it itself it benefits from the food that is taken by the human being or the animal within their bodies. So there is a mutual benefit between the body and the bacteria. So this we call them commensals, and usually we call them microbiota. Microbiota, that means the normal bacteria that is found in the body of human being or in animals. And the science dealing with this normal bacteria within the body, we call them microbiome study, or microbiome. It's a collective name for this bacteria found normally as commensals in the body of animals and human beings. While pathogenic bacteria, these are bacteria which causes disease. Some commensals, they can revert, they can change, convert from being commensals or non causing disease, but change for some reason or the other. Maybe the immunity of the human being becomes very weak, or the animal, or there is a change in the habit of the food of the human being or the animal, that lead to this bacteria become change from commensal into pathogenic, causing disease. But at the end, pathogenic bacteria, it's a type of bacteria that causes disease within this human being or an animal body. This pathogenic bacteria, it could be, as I mentioned, a commensal, or could be a bacteria that is from outsider, from the environment where the human being and the animal lives. So we have two types, commensals and pathogenic bacteria. And definitely, if there is a reservoir, there is a habitat. Reservoir, that means there is a habitat for this bacteria, either it is commensal or pathogenic. If it's, for example, the bacteria enterobacteria or Escherichia coli, it is found in the gut of the human being. And this, the gut, we call it as the habitat or the reservoir where the bacteria lives. But at the one point, it could change from commensal into pathogenic for different reasons. Either it change its genetic makeup, or maybe changing the food of the human being, or change in the environment, pH where it lives. So there are different factors. So that's why we need to speak about environmental reservoirs. And these are generally divided into indigenous, that is means inside the human body or the animal body, or exogenous environmental reservoir, and this somewhere in the environment. Maybe a dog, maybe a cat, maybe a type of a soil, maybe different other, in maybe a water. This is where the bacteria lives its normal life. Once it enters the human body, then it changes to be harmful and causes a disease. That is why I'm grouping here the different tables to show the different types of bacteria that causes a disease. And these tables are very important for you to note and know by heart, especially the name of the bacteria, the morphology of the bacteria, the reservoir, and the type of infection. These four columns are very important. The name of the bacteria, the morphology of the bacteria, the reservoir, okay, and the type of infection. Of course, while you are studying, you will be able to note if it is a commensal or not a commensal. That means it lives normally within the human being or not. But of course, from the reservoir, you will be able to note if it is commensal or not. So we have three tables depending on gram stain, gram positive bacteria. For example, I will take one example to explain the table, and then you can follow the rest. For example, in gram positive bacteria, if we take like listeria, for example, listeria is rod shaped. Okay, it's not a commensal. Okay, and of course, as you can see here, it is rod shaped. It is fermenting sugars. This is the requirement of for oxygen requirement from fermentation, and but it is a pathogenic because it is not a commensal, and at the same time it causes a disease. Okay, meningitis, that is suhai as we call. So this is an example of a bacteria. It causes a disease in human beings. For example, if we take another type of bacteria, for example the staphylococci, these are cocci or grape shaped. Okay, it is fermentive, aerobic, and it is a commensal, and it is found in the on the body, okay, on the skin and the nose and so on. But it can cause disease in the human being, changing from commensal to be a pathogenic, causing disease in the human being. So this is how you read the table. Also, you have another table for gram-negative bacteria. For example, if we take Vibrio cholera, because this is very common now in Sudan after the war, and this is caused cholera. So it is rods but curved in shape, and this is microrephilic for oxygen requirement. That means it lives in carbon dioxide, a medium or an environment that is full of carbon dioxide. It is not a commensal. The reservoir, it is water contaminated with this, or contaminated food and water, and causes diarrhea. So this is an example, and so on. You can look into the other type of bacteria, but you need to know these tables. The third table is the miscellaneous bacteria. As you know, there are certain type of bacteria which does not have a cell wall, or they have a cell wall which cannot be identified by gram stain because it's not the same structure of a normal cell wall, bacterial cell wall. So, for example, if we take treponema, it is it is not visible in the gram stain because of the cell wall. It doesn't have a cell wall, and it's not a commensal. Okay, and it transmits to human beings by sexual intercourse or contaminated bloods and so on, or contaminated human extracts. And so this type of bacteria causes what a disease called syphilis. So to this end, I end this session, and wish you all the best.